Good evening, everyone. My name is Danielle Hake, and I'm the RiverWatch Director and Stream Ecologist with the National Great Rivers Research and Education Center. For those who are new to the Illinois RiverWatch Network, RiverWatch is a citizen science organization that trains volunteers to monitor water quality in their own communities. The mission of RiverWatch is to safeguard the future of Illinois rivers and streams through stewardship, education, and sound science. And before we get started, I'll ask you to keep your microphones muted during the presentation to help limit the distractions. There will be a Q&A session following the presentation. So if you have a question you would like to ask, you can either put it in the chat box and the presenters will answer it at the end of the talk, or you can save it to ask aloud during the Q&A session. And now I would like to welcome our speakers, Hannah Griffis and MaKenzy Sorsen. Hannah graduated with her master's degree in biology from Southern Illinois University in Edwardsville in 2020. She is currently working as a RiverWatch technician for the program. MaKenzy Sorsen is an undergraduate student at SIUE. She will be graduating with her degree in biology focused on ecology, evolution, and conservation. She worked with us this past summer as an intern, and she studied the distribution of Asian clams in the St. Louis region. So our presentation will begin with Hannah giving us a background of invasive species, and then MaKenzy will present work from her summer internship with NGRAC.
All right, thank you, Danielle. So like Danielle said, I will start by giving a short intro on invasive species, and then MaKenzy will talk about her work that she did. So invasive species, this definition here comes from the Invasive Species Executive Order from 1999, and it has two requirements for something to be considered an invasive species. The first is that a species is introduced to the ecosystem, and this just means that the species was, sorry, I just realized that I can't see my notes and I thought that I would, and I have a lot of stats that I wanted to add in. And Danielle, do you know how I can see my notes? She's gone. Okay, I'll just keep.
So yeah, usually I do a split screen, so I have the two monitors, so that works. But
because when I get out of my PowerPoint, I think you guys see that I'm out of my PowerPoint. So
okay.
Well, oh well. Okay, anyways, sorry. So two requirements to be an invasive species. One is that they're non-native to the ecosystem. So this means that they were not originally in the ecosystem, and they somehow moved into the ecosystem from either their dispersal or from human actions. This doesn't necessarily mean that they came from another country or something like this. This can happen within a country or a smaller area. For example, lake trout are native in the Great Lakes, but they are considered I'm sorry if you guys hear my cat playing also, I'm just all over the place here. But okay, lake trout are native in the Great Lakes, but they're also found in Lake Yellowstone in Wyoming, and they're considered invasive there. So it doesn't have to be out of the country for something to be considered invasive. The second thing that has to happen for a species to be considered invasive is that it has to cause or is likely to cause in the future harm to the environment, economy, or human health. And we'll get into that later. All of the pictures that I have on this slide are invasive species that occur in Illinois, and I'm sure a lot of you have heard of most of these. I tried to pick common ones for the slide. So there's a few different pathways on how invasive species will spread from one place to another. They can either spread through natural pathways, and this is like wind, currents, or any sort of way that a species naturally disperses is considered a natural pathway. And then they can also be spread through man-made pathways, and this is just any activity that is either created or enhanced by human activity. So this is anything that a human has caused the spread of a species. The man-made pathways are either intentional or unintentional. The intentional pathways are things that we as humans are directly doing to move a species from one place to another, like on purpose. One thing that happens is for pest control. We use biological controls sometimes, so we'll bring in another species of animals to control a pest that we're having problems with. So like a predator of the pests, and sometimes the species we bring in end up causing harm to the environment. The pet trade, so pets are often released either by accident or on purpose because somebody took in a pet that they couldn't handle, or they just decide that they don't want it anymore, so they just release it into the wild. And then importing plants, a lot of non-native plants are planted just because they look pretty, and then they end up spreading into wild populations and can take over or lose control of their, the population just spreads faster. And then you can have unintentional ways of spreading invasive species. So a lot of this happens with our transporting methods. So species can be moved with ships or boats. So aquatic species get spread this way. They can be spread when we're transporting wood. So this includes like firewood or even crates or boxes. And that wood can have pests or insects that get transported with them. And then again, with importing plants, those non-native plants or even native plants, if they're maybe traveling long distance or being put into a new environment, they could have insects that are living on them or a disease, and that can spread into a new environment. Okay, so at the beginning, I mentioned that the second part of the definition, the species have to cause harm to either the economy, human health, or the environment. So we're going to go into those three things in detail, and I have some examples. So for the economy, globally, for the past 50 years, we have spent $1.288 trillion on invasive species. And this includes a multiple of things that are all listed there. So property values can decrease from invasive species. We can have less agricultural and fishery productivity. They can cause problems in public utility operations. They can also cause a decrease in tourism or outdoor recreation. And then this also includes any of the methods that we've spent money on trying to control invasive species. So I have two examples here of species that have had an impact on the economy. Sea lampreys, in their native habitat, they act as parasites with another host species, and they don't usually kill their host in their native habitat because they co-evolved together. And so sea lampreys have been imported into the Great Lakes. So they're not native to the Great Lakes, and now they're there. And because they don't have that co-evolutionary relationship with the species that are already living in the Great Lakes, now they act more as predators. And I believe the stat I had for this one was that they consume up to 40 pounds of fish in their 12 to 18 week feeding period. And this has been really bad for the commercial fish populations and for fisheries. And then zebra mussels, I know a lot of you have probably heard about. Zebra mussels will attached to hard material and really pile up. And so you can see this picture of a shopping cart that's just covered in zebra mussels. So this can happen on ships, and they'll cover like the propellers of ships. And it can be really costly to have them removed and to fix the repairs afterwards. And this can also happen in pipes, and they'll block water intake pipes. And so that's also costly for like power plants and cities to get those removed. So invasive species can also have impacts on human health. So they can infect humans with new diseases, and then they can also act as vectors for existing diseases, and that makes the existing diseases worse in certain areas. And then they can also cause wounds to humans. So that includes bites, stings, allergens, and toxins. The two examples I have for this one are pretty popular, especially like in the news. Asian tiger mosquitoes get spread a lot with international used tire trade. And they act as vectors for a lot of viruses, and that includes dengue fever and West Nile virus. And then the Africanized honeybee, it is a lot more aggressive than the European honeybee that we're used to seeing. And it is more likely to swarm and to attack humans and animals. And it's also been made into a lot of horror movies, so you can tell that it is more aggressive than a normal honeybee. And then finally, we have the impacts on the environment. So the invasive species can either be directly harmful to the native species, or they can harm the ecosystem in general. And the impacts on the environment are either direct or indirect. So the direct impacts include predation, herbivory, and disease. And this is basically anything that the invasive species are doing to directly harm or kill other species. That would be a direct impact. And my example here is domestic cats. And I'm not coming after anybody who owns cats, because obviously I own a cat. You can hear him probably in the background. But domestic cats kill a lot of animals. I had numbers for this, but they kill more than 1 billion birds a year, and then like more than 3 billion mammals a year, I think were the numbers. And they have also been, the American Bird Conservancy has estimated that they have been involved in the extinction of 60 animals. So domestic cats can be a really big problem, especially the feral cats. And then indirect impacts on the environment include things like resource competition. So this is just when species are outcompeting, the invasive species are outcompeting the native species. So they're either better at getting food or better at using the resources in the environment or things like that. And then also habitat alteration. So this could be anything from changing the plant makeup to changing the soil chemistry and the water chemistry. And this can all affect other species in the area. So my example for an indirect environmental impact is kudzu, which is a vine. It's a big issue in the southeastern Illinois or southeastern United States, and it's starting to move into the Midwest. And the vine pretty much outcompetes every other plant in the area because it just covers them. So if you can see in the picture, the vines are literally covering the trees. So all the plants that are covered can't photosynthesize. So it really lowers the biodiversity in the area, which can also impact not just the plants, but the animals and the insects that use the native plants. Okay, so now we kind of have a background on invasive species. I'm going to talk about Asian clams a little bit. So we know that Asian clams are an invasive species. They are native to the freshwater environments of Eastern Asia and Africa. They are hermaphrodites, so they produce both sperm and egg at the same time. And this means that they can cross-fertilize or they can self-fertilize, which also means that it only takes one clam to start a new population. They live about 3 or 4 years, and they usually breed twice a year, and they can produce 1,000 to 100,000 juveniles, which they do have a really high mortality rate, but you can see how one single clam could potentially start a new population in a new area. If you were at the last speaker series, they talked about how native mussels use like a fish host or a salamander to as part of their reproductive cycle, and they use it as a host for their juveniles to grow on. Asian clams do not do that. They fertilize the egg internally, and then all the eggs sit on the gills of the Asian clam for about 4 to 5 days. And then they are released into the water, and they have, they can disperse really long distances, either through water currents or by catching a ride on other animals. And then once they are adults, they are about 10 to 30 millimeters for their size. And then you can either find them slightly buried in the sediment or sitting on top of the sediment, and they act as filter feeders and remove organic material from the water. They're pretty widespread in the United States. So on the map, the dark red is where the Asian clams have been found in the United States. And so you can see that includes Illinois and Missouri. They're not entirely sure how Asian clams are spreading in North America, but the main suggestions are human transport, so being spread through boats. And then they're used in aquariums, so they could be let free from people who get rid of their aquariums. And then also through the water current, because they're able to just ride that long distances to spread. And their impacts are mostly ecological and on the economy. So for the impacts on the ecology or the ecosystem, they outcompete other filter feeding species. So they're really good at taking in nutrients, and they're really efficient at using the nutrients around them. So they can take up food from other species. And then they also change the water quality and the substrate makeup. So when there's really big groups of Asian clams, they will release a lot of nitrogen. Sorry. Okay, sorry, I was being bit by a cat. Okay, so they really release a lot of nitrogen, and they also, in their native habitat, they're not really used, like they don't see extreme temperatures. So when they're in these new habitats and they hit extreme temperatures that they can't survive, there'll be mass dying events. And then when this happens, a lot of nutrients is released from them. And then there's also a mass decomposition event. And both of those things can be really bad for water quality. And then also after they die, their shells just become part of the substrate, and that can really change the makeup of the substrate. So if the substrate is just all silt and sand, and then suddenly there's a bunch of shells mixed into the substrate, it can attract different species. For example, zebra mussels really like hard substrates, so they can move into areas where there's a bunch of Asian clam shells. And then for their impacts on the economy, they can disrupt pipes similar to zebra mussels, but they don't hold on to hard surfaces like zebra mussels do. Instead, their juveniles can fit through some of the screens and filters that are on the pipes, and then they'll settle in where the sediment has settled in the pipe, and then they'll reproduce, and every time they reproduce, they move deeper and deeper into the pipes. And this can eventually clog the pipes. It can alter the flow of water in the pipes, and can also lead to more sediment building up in the pipes. And then they can also deter recreation and tourism. So you can see in this picture on the beach, there's a ton of shells, which these ones are kind of colorful, but I feel like most Asian clams after they're dead are mostly like black and white and not very pretty to look at. And then I imagine this also wouldn't be very fun to walk on. Okay, so now that we have a little bit of background on invasive species and Asian clams, Mackenzie is going to share her work that she did this past summer.
All right, thank you, Hannah. Let me get this pulled up. No, I can't see. All right. So my work was focusing on populations within the St. Louis region. So we mainly focused on the Alton area and then the Webster Groves area, and then some more in the northern area of St. Louis County. So as sort of an extra introduction as to how they came to be invasive in our area, they were first found in our area in the 1920s, and it is believed that they were brought over either by ballast waters and ships like Hannah had talked about, or as a food source from Asian immigrants that immigrated here. Currently, they are found in 85 of 114 counties in Missouri and 93 of 102 counties in Illinois. And they were first found in our area, like the Mississippi River, in the 1970s. So as an invasive species, they can be extremely detrimental to the native mussels in the habitat. They're so detrimental to these species due to the fact that they filter feed at a much higher rate than any other mussel does. And when filtering this out, they are filtering out both other mussel sperm and their food. So these other species are having a double whammy where they aren't able to reproduce or grow to reproductive size. And removing them can be incredibly difficult as well as costly. There have been several attempts to do so. A few in Lake Tahoe, they put like mats on the bottom to kind of prevent them from being able to get food and stuff. And then also they went around and suctioned up as many as they could. But they found that this was ineffective. They also tried to heat the benthic layer in Lake Tahoe, and that was also a failed attempt. The most effective method that they've used was only effective short term, and that was with the benthic barriers in Lake George. And they found that it killed off the adults, but juveniles were able to escape the barrier. So eventually the populations would bounce back due to there still being juveniles. But they did find that these methods, especially the benthic barriers, did not affect the other benthic vertebrates, or invertebrates, sorry. And just this week, a California representative proposed a new law to try to prevent the spread of the mussels, of invasive mussels. So specifically, they were talking about quagga mussels and zebra mussels, which are, as far as I can tell, pretty much the same thing. There's just like a super tiny difference. But it would also include Asian clams in the act. And so it would allow the federal land management agencies to inspect their watercrafts before coming into a protected area or leaving an area, to search their propellers or the trailer for stray mussels and clams. And this is really the only way that we will be able to stop the spread of it, or at least limit it, because this is one of the main ways that they spread. Especially if you have a lake that is cut off from everything else. The only way that they're going to get back to that area is through human transport. So our objective of our study was to find the populations, determine if there were populations, and whether or not that population that we found was a source or sink population. A source population is a population that is thriving and contributes to other populations in a different area. A sink population is where those extras go, and they're not necessarily thriving, they're just existing there. So that population doesn't necessarily grow. So to start out with, our methods, whenever we got to a creek or a stream, we would set out a line with, it had different knots on it at different lengths. And then we would choose 5 spots along that rope, trying to get as varied sediment types as possible. And also pools or riffles if that was possible. And then after we chose the sites that we were going to be sampling, we used a PVC square, and we would hold it down, and then we would dig 6 inches into the sediment. And that sediment would be placed into this green sieve that was 4 millimeters in size. And what went through would be caught in the bucket so that we could re-sieve it in a smaller sieve. And what was caught in the green sieve, if there were clams, would be considered adults because of their size. And what fell through, if anything, would be considered juveniles. So here are examples of what we found in our large sieve. It really varied depending on our sites. We were in Missouri and Illinois creeks, which are incredibly different. This is an Illinois site, and you can see it's pretty sandy, even a little bit of silt, a little bit of gravel, but not much. And then we'd go to a Missouri site, and it would be bedrock and giant boulders and huge rocks. So that varied incredibly. So this is from an Illinois site, not much there. And then this is also from an Illinois site, but that is a muddier site and siltier. So once we sieved it through the green and inspected it, then we'd use, this gold one was a 10 micron, which ended up being too small. We switched to a 20 micron, I believe, but still super small. The clams that Hannah was finding were like as big as the tip of a pencil. They were incredibly tiny. Once we did find clams, we placed them in a white tray with water in it. And we would wait to see if they came out to make sure that they were alive, to ensure that we were only counting live ones, at least for their size. And then once their foot came out and we saw that they were moving a little bit, we would use a caliper to measure the ventral width and length. And we would also do counts of live adults, live juveniles, juvenile shells, and adult shells. So these two here are shells. And you can see how tiny this one is compared to all the other ones. So once we were done in the field, I came back, and while in the field, we took water samples as well as sediment samples. So with the water, I would use this contraption to filter out water. These cups are magnetic, and you put a piece of filter paper over this, and then it filters out any particles like sand particles and stuff like that, so you can get a better reading. This is a total carbon calculator and total dissolved nitrogen calculator. We found those to not be super helpful. Everything was pretty much the same in regards to that. There was no obvious patterns. And this one calculated dissolved nitrogen and phosphates and nitrates. So with the sediment, I would dry it. That could take anywhere from overnight to, I think one took over a week. Felt like forever. And then after they were dried, I would put them through all these sieves and break them out into fractions to see what fraction was coarse sand, gravel, fine sand, medium sand, silt, that type of stuff. That also took forever. I think I spent a full 2 weeks doing that alone. And so these were our total numbers for what we found to be alive. So the highlighted ones are not alive, but just found in general. The highlighted ones were ones that we found live clams at. Some we found no signs of clam life at all. Others we found a few shells. Some we found lots of shells, but no live ones, which was weird. So starting here, these ones are Missouri streams, and then over here are Illinois streams. So this is just a map of those sites. So the red dots are where we found live ones. So you can see that we found one live one in all of Illinois, or one site that had live ones in all of Illinois. And then the rest were in the Webster Groves area. But also in the Webster Grove areas, there were areas where we found no sign of clams at all. And then up here in the northern part of the map, we found some signs of shells and whatnot. But the further away from the Mississippi you got, you found less and less signs of them. And although we did not sample the Mississippi using our methods, because it would have been impossible to use our methods on the Mississippi, we did go down and observe shells of reproductive size, which was the 10 millimeters. So that is a population also within the Mississippi River. This is the sediment makeup. So here you can see I took 2 sediment samples at each site, so that's what the number 1s and the number 2s are. So you can see that a lot of the ones that we found clams in did have gravel, whereas other ones didn't have a lot of gravel. Yep. So here our sand was somewhat significant. The more sand that you had, the less, so it's this blue line right here. The higher percentage of sand that you had, the lower the chances of finding clams were, which was contradictory to what I had read in a lot of the literature. And then here the silt was significant. But this could have been skewed due to the amount of sites that we had that were plotted at 0, 0. But it looks what the line means. It's the black line. The more percentage of silt that you had, the higher the number of clams that you would find were. So in conclusion, we had, I don't know if this picture matters. So a lot of sites we couldn't determine whether they were a source or a sink population because there wasn't enough data. Like if we found nothing, we couldn't say that it wasn't a population because we only looked at 5 sites that were a square, not even a square foot in diameter perimeter. So that's why there's a lot of gray up here. That's where we didn't find a whole lot. The one that we did find over here led us to believe that this area would be a sink population. This area here off of the Missouri River is also likely a sink. And then we couldn't determine this population. And then these ones over here were likely sources due to the amount of clams that we found. So them being source populations is not a good thing because that means that they are able to reproduce enough to create populations in other areas. Another thing that we found while in the Missouri streams was that we would find piles and piles of open shells, and it looked like something like a raccoon or possum had eaten them. So when we found areas like this one up top with the large rocks, we would find a lot more live clams underneath the rocks. So we thought that predation played a large role in the type of habitats that the clams would choose to live in. And yeah, that's what I got. Doing pretty good on time, I think. Danielle?
All right. Thank you, Mackenzie. And thank you, Hannah. Let's go ahead, and I got the chat open here. We have a question already from John Griffis. Do Asian clams have glochidia that attach to the fish gills? If not, do the larvae filter feed as well?
I can answer this because I feel like it was part of my introduction. They do not use fish as a host for their larvae. They fertilize the eggs inside of themselves, and then the eggs sit on the gills in like nutrient-rich water until they're big enough and ready to go out into the world. And so I would assume that when they go out into the world, they are ready to filter feed.
Right. Any other questions? While other people are thinking a little bit, I have a question. So Mackenzie, I maybe I missed this, but so how, for that last, for that map, how did you decide which populations were likely source versus likely sink?
So we determined that by whether or not we had found more than 3 live clams in an area. I think 3 was our magic number. And if there had been shells and like 1 or 2, then I think we called that one a likely source. Whereas if we only found shells or a very small population, we called it a sink.
All right, another question from John Griffis. Do you know if there's any correlation between stream quality and the numbers of Asian clams?
So I'm gonna be totally honest, we did not find a lot of clams in a lot of sites. That was a big issue, especially in the beginning. I got very discouraged early on because in the Illinois streams, we found 1 or 2, and that was the only sites that we had sampled yet. But with that being said, all of the water quality parameters that we ran in regards to water quality, temperature, sediment types, that type of thing, they did not have an effect on it, but we did not find many large populations. So I would feel more comfortable saying a definitive answer if we had found more populations that were larger. I think we even kind of did diversity with what we had found a little bit with what we saw, and that didn't correlate either with the number of clams.
Like the diversity of the other animals that we saw while we were there. Okay.
Yeah.
Incidental observations.
The diversity of me picking up rocks and telling you what's on them.
Yes. And scaring me with.
Okay, we have one from Haley. As a novice naturalist myself living on the DuPage River, what is the best takeaway to avoid spreading or decreasing the spread in my waterway? Other than managing my watercraft, a kayak, what can I do?
Okay, so definitely check your kayak that you're taking in and out. I would even spray it off because, like I said, the sizes that we were finding were incredibly tiny. And the same thing with the shoes that you're wearing, making sure there's none stuck in the crevices of those. Missouri told us that if we found live clams, to throw them out on the shore to limit their population sizes. So if you feel comfortable doing that, you can do that. I personally didn't like doing it, but yeah, there's not a lot that we can do other than just watching what you're doing. Not contaminating is a big thing. If you're going from one area to another, clean your stuff, and that's the biggest thing that you can do.
I had one other quick question. Do the larvae actually physically attach on boats and such?
So I don't know. I know
zebra mussels do.
the zebra mussels do emit a slime that makes them sticky. And we did see them spitting out slime, but I don't know that it was sticky, like in order for them to stick to things. Like they wouldn't stick to the rocks that we were picking up. They weren't on the bottom of those. They were just in the sediment. So I don't know that they would necessarily stick to the boats rather than just be in, like on the inside in little, like the bottom of your boat where it catches some water sometimes. I think that's where they would be at. I don't think they'd be on the outside. But zebra mussels are a huge issue too, and those can definitely get stuck on the outside.
Okay, thanks.
All right, do we have any other questions? Looks like that might be it. Well, thank you both thank you both very much for your presentation tonight. And then actually, Hannah, do you have in front of you what our next, it was on the rotating slides earlier, but
Life along the Mississippi before 1942. So it's going to be an anthropology one. And she said she's going to focus on the ecology. So like what they use the river for. I'm not entirely sure what she's going to talk about, but she was one of my teachers and she's a really good speaker. So, and that's on November 4th. So next Thursday.
okay, great. Well, we hope to see a bunch of you again next Thursday. And until then, be sure to wash your boat before you move it to the body. Have a good night.
Thank you.